you. It's terrific to be here. Can you all hear me? Yep. Okay, great. So this has been my sort of favorite talk title to use recently. The cell is a bag of RNA. It's meant to be a little bit provocative. And it's also meant to evoke sort of a historical resonance of this battle between biochemists and cell biologists about whether or not you can think about the cell as a bag of enzymes, which is the way the biochemists think about it. And the cell biologists say, no, you have to think about all the complex structures and compartments and so forth. And this is the history of that phrase, bag of enzymes. It dates back to the 1930s or 40s. And you can see the battle has waxed and waned over time. And of course, the cell biologists are right at sort of some level that those structures really do matter and the cell can't do what it does without them. But on the other hand, biochemistry, by using that simplifying abstraction, has had such a powerful impact on our understanding of how cell works that you can see it's useful to make these sorts of abstractions and then revisit the full complex problem later. And that's been sort of the approach I and many other people in this meeting, I think, have pursued with respect to single cell biology, which is just, let's use these powerful tools we have to look at RNA and enumerate all the RNA molecules in the cell and just imagine it's a bag of RNA and forget about everything else for a little bit and see what we can learn from that. And I'll take you through some examples of that. First ones, I'll just highlight a few recently published things and then get into some unpublished work in the latter half of the talk. So, it's been appreciated for a very long time that even in a phenomenon as complex as memory, that RNA and RNA synthesis plays an important, crucial role. And in fact, these were some very early single-cell transcriptome experiments done by Kendall and collaborators to work through that and discover some of the genes involved. Tom Sudoff and I decided to try to revisit this program using a more complex model, mouse, instead of slug, C-slug, and which has a sort of more sophisticated form of memory and the powerful tools of single-cell transcriptomics. And we did that using fear aversion training. The mouse is put in a cage and a tone is played and gets a shock and it learns to associate that sound with something unpleasant. And then you can play the sound and the mouse freezes because he or she recalls the unpleasant behavior, consequence, stimulus. And we've used the very powerful TRAP2 mouse developed by Leach and Low to try to encode which neurons are being activated when the mouse recalls its memory well after the stimulus. And when I say we, I mean two very talented postdocs from our labs, Shan Jiang in Tom's lab and Michelle Chen in my lab. And so we're doing the recall more than 2 weeks after the memory was formed. So this is really long-term memory formation and recall. And the cells that are involved in the recall are made permanently fluorescent. We then analyze them by single-cell transcriptomics. And sure enough, we're able to find very clear differentially expressed genes and sustained transcriptional signatures well after the event. As you sort of dig through those genes and pathways and classes, ontology classes, you find, for example, very clear view of vesicle exocytosis, which is upregulated in those cells, and as well as many other interesting effects that I don't have time to do justice to here, but wanted to sort of flag this as one example of how one uses this approach in conjunction with all the other powerful tools of cell biology to look at a really complex behavior. As another example, and here just a single slide on this paper, this is work done between my lab and Leach and Low's lab and a bunch of other folks involved as well. But an attempt to use single-cell transcriptomics to understand the evolution of anatomical structures in the brain, in this case, the cerebellar nuclei, which are part of the cerebellum. And we did comparative studies in chicken, mouse, and human, and found that it had been known that these structures are present in different numbers in these species, in invertebrates. And we were able to show that these evolved by having conserved cell types and duplicating those over time. And this is, I think, an example of what's going to become a very productive research area in the coming years of using these single-cell transcriptomic atlases to understand evolution at the level of cell types and the role of cell types in evolution. And I think there's a great future in that. So now, let me tell you, so those were things that sort of done in my lab as regular science projects between collaborations between two groups. Let me tell you now about some of the larger projects we've embarked on using the resources of the BioHub, which have involved many different labs across institutions to do things that are sort of what would be difficult to do in any one lab or project or grant. And so we began exploring the idea of establishing organism-wide cell atlases and thinking about them as references that would be companions to the genome that will help you understand how genes are used in various cell types. And we began in the mouse. We called it the Tabula Muris, and then the Tabula Muris Senis, the mouse aging cell atlas. And this is work that was led by myself and Tony Whisker-Ray, and at the BioHub involves Spiros Damanis and Angela Pisco, and many, many other groups at Stanford and UCSF and the BioHub, and I'll acknowledge some of them as we go on. And here we looked at something like 18 different tissues, looked at roughly 500,000 cells over them, and looked at them at various ages of the mouse, including out to 30 months, which is like a human centenarian. And part of what we did here was use a very small number of mice because we took all the tissues from the same mouse, and we worked very hard on the coordination so that these essentially the tissue-to-tissue comparisons were perfectly controlled for age, epigenetics, environmental effects, and so forth, because they're all taken from the same mouse. And that became a theme of how we've approached these atlases. And so to do that, it involved, as I said, a lot of coordination. This group of, we involved tissue experts, labs that were experts in each of the tissues we were studying. The student or postdoc from that lab all showed up, ice bucket in the hand every morning. A mouse was sacrificed, and they all took their organs back to their labs, did the dissociations and the sorting, brought them back to the BioHub, where all the cDNA was made and the sequencing was done. And the leaders of the sort of BioHub platforms there were Norma Neff on genomics and Jim Carcanius in data science. And it was, as I said, a lot of coordination, but a lot of fun to do. It's all published, so I'll just, 2 slides on the data here. One is the ability to look at aging-related effects in shared cell types or shared cell states across tissues, in this case, senescent cells, able to see that number of senescent cells is increasing with time, and the expression of key genes involved in senescence, like CDKN2A, is increasing as well. And more generally, as a tool to kind of, these single cell atlases are a tool to deconvolve effects that get mixed up in bulk measurements. Like, is it the number of cells expressing the gene that's changing, or is it the level of expression of the gene that's changing? And we can, with single cell approaches, can separate those two effects, and have done that more generally in this work. Also, we've been able to look at not just gene expression, but because some of the data was taken with SMART-Seq2, able to have whole transcript sequencing and to look at mutational load and how that changed with age, and look at that in comparison with RNA spike-ins. The third paper in this series of mouse atlases is not published yet, but it's available in bioRxiv. And this is a functional study where Tony and Tom Rando spent years kind of exploring this, have stitched together the vasculature of 2 mice. That's called parabiosis, when 2 mice share the same blood supply because their vasculature is attached. And if you do that with mice that are of different ages, it's been observed that the old mouse starts to appear younger, and the young mouse starts to appear older. So there's a rejuvenation for the old mouse and accelerated aging for the younger one due to this shared blood supply. And the reasons for it have been unknown, and we decided to try to understand some of the molecular basis of this by building an atlas of these parabiosed mice. And so we did it again with a large number of tissues, and because we have the reference of natural aging, we can now compare accelerated aging with natural aging and ask, is that similar? And we can compare rejuvenation to aging and say, are things really getting reversed? And we can do that at the level of many different cell types across tissues and number of different genes that are differentially expressed with aging. This is like a master table that kind of shows it. The little scale is the key is at the bottom. The things on the left side of the circle are the accelerated aging. On the right side are rejuvenation. And blue is consistent with regular aging. Red is not consistent. You can see many of these differential genes, especially the ones that are downregulated with age, are consistently reproduced both with rejuvenation and accelerated aging. Whereas upregulation is a little more mixed and not as clearly consistent with natural aging when you have this parabiose model. But to give you one sort of bottom line story here, as you might expect, energy matters here, and genes involved in electron transport in mitochondria are predominantly downregulated in age across many, but not all cell types, but the vast majority they are. And these are ones that are well reproduced, both with the accelerated aging, and you see it going the other way with the rejuvenation. And so there are some broad themes that are unfolding from this. And again, I'll refer you to the preprint if you'd like more details. And in the remaining bit, I'll walk you through some of the other large-scale efforts we've undertaken collaboratively to build these reference atlases. We're approached by the folks in the Drosophila community who were looking for some assistance in organizing an effort to do a fly atlas, and we've been part of that. And this involves a number of different labs around the world. So the overall coordination is Leach and Lowe, Norbert Perrimon, David Sutherland, Stein Ertz, Bart DePlanck, and myself, and Hongji Lee, who just was a postdoc in Leachen's lab and just became independent faculty member at Baylor. And we've involved, again, tissue experts to do tissue-by-tissue analysis of the flies. It involves 13 labs from all over. We've done most of the sequencing at the BioHub, some at the NIH, and the data processing and analysis has been led by Stein and Bart's groups, and they've been great about bringing in the larger community to do annotation jamborees. And so, again, like our mouse data, a huge number of these cells have been hand annotated by experts, and people have really thought about what the cell types are in the context of existing cell biology knowledge, and thereby provide a lot of sort of added content. This is one example of one tissue, body wall, thorax and abdomen. And you can see that cell types that had previously been identified by just 1 or 2 or 3 genes now have entire transcriptomes associated with them and allow a much deeper understanding of what's going on. We've got, this is an older one, this is 250,000, a little higher than that now, pushing 400,000, I think now, cells with some form of annotation. And a couple hundred different cell types that have been really carefully looked at. And we think this is going to be a great resource for the fly community going forward. Data is all taken, papers being written, almost analysis is being done. So you can look forward to this appearing on bioRxiv and having the data released sometime in the near future. Another one we've been working on, and which is close to completion, is a non-human primate, the mouse lemur. Lemurs are very interesting primates found in Madagascar. The mouse lemur is on the evolutionary scale almost exactly in between mouse and human, at the primate, but it's very small, and it has many properties of a model organism like the mouse does that make it attractive to work with. And my colleague at Stanford, Mark Krasnow, has spent many years trying to develop it as a model organism, and he was our lung expert on our mouse cell atlas, and convinced us that we should use the tissue expert teams to also do a lemur cell atlas, because he has a colony at Stanford, and the lemurs occasionally have to be sacrificed due to old age. The vets say it's time to put them down at some point, and that happened a few times recently, and so we pulled a group together to build a lemur cell atlas. About 250,000 cells, 30 different tissues, and the first non-human primate cell atlases. And again, there's been an enormous amount of effort put into hand annotating the cell types. And again, paper is almost fully written, and so you should look forward to seeing this appear again in the not too distant future. We're very excited about it. It's been an enormous amount of effort put into and trying to understand the various cell types and connect them to what we know about human and mouse cell types, returning to this theme of looking at evolution. And there's some very interesting cell types that appear that the lemur shares with human but not with mouse, a couple in the lung, for example, as well as other tissues. And then finally, we're working on a human project as well, following the same sort of philosophy where we're trying to obtain something like 20 to 25 tissues from the same individual donor, so that you can look at shared cell types and have it perfectly controlled for genetic differences, environment, age, so forth and so on. And this is led by Bob Jones in my lab, and involves, again, a very large number of people across all 3 institutions, 3 universities in the Bay Area, as well as the BioHub. We're just kind of at the beginning of this. We've done a few practice. We've been working closely with one of the organ procurement operations that finds donors for transplant, and have been doing some practice attempts. And we've done a pilot with 6 organs, a pilot with 18 organs from the same individual. We just did one with 20, and that data is on the sequencer right now. We've done many of the organs individually, so from different donors, but we've sort of got it all kind of coordinated and working now, and we're pretty excited about what this will serve as a resource going forward. And I'll just sort of close by saying, as I've tried to emphasize, these are really team efforts involving large numbers of people, faculty, students, postdocs, staff members. These are pictures of some of the folks who are involved in mouse and lemur, and with human, it's an even bigger list. These are many of the tissue experts involved. In the picture, you see some of the core team members who are there pulling all-nighters when the donor's available. It's kind of a real complicated dance, and it's something because we're working with the intact cells and not the nuclei, we're doing it all in a pretty narrow time window, and have got that working pretty well right now, and thanks to the efforts of these folks. So I'll stop here, and happy to take any questions, and just return to this notion that this abstraction of thinking about the cell as a bag of RNA is a very powerful tool for understanding cellular identity, and we think these large-scale atlases are going to be super powerful companions to the genome and understanding how different cell types and tissues use the genome. Thank you.
Thanks very much, Steve, for an excellent talk. I was happy to see the small and test time on the new atlases. I was using them in the Tabula Muris, so that's great.
Terrific.
So we have a few questions. One from Zhu, from the young age parabiosis dataset, could you tell if certain tissue ages rejuvenate earlier than the others?
Yes, there's very interesting data on this kind of time course effect and of aging. And the strength of the effect. And I'll refer you to the preprint there to have a look at it. And not all rejuvenate. It's not uniform, right? It definitely effect size changes. Sorry, go ahead. The next question.
I'll ask a different question. Very impressive atlases. This is from Homaira Hamidzada. In these large atlases, how do you go about ensuring robust reproducibility? Besides pooling several tissues, would it be feasible to reproduce in a different lab and assess the stability of gene signatures for each cell type population?
Yeah, that's a very good question. So, of course, we're doing biological replicates, and that's one way to assess reproducibility. Another way is, we've been kind of concerned about the systematic errors involved in the measurement. And so I alluded to this briefly, but I'll expand on it now. We've tended to use 2 different technical approaches to do the transcriptomes. And so, for example, with Tabula, with all of them, well, no, not with all of them, but with the mouse, for example, we've used both 10x and SmartSeq2 to obtain transcriptome data, and we can try to understand what are the kind of technical artifacts that each of those methods introduces by comparing the data sets from both of those and looking what's shared and what's not.
Thanks. We have another question from John Murray. Any plans for developmental atlas generation in interesting comparative organisms like mouse lemur?
I'm sorry, what was the question?
So whether you're planning to do a developmental atlas
developmental
organisms
atlas.
like mouse lemur.
Yeah, not with the lemur, because we can't, ethically sacrifice them at young ages. There we're being opportunistic, and we're only working with them when the vets say it's time to put them down. But with the mouse, obviously we have, and we also with the fly have an aging series as well. That's coming after the first fly atlas, but we're working on a fly aging series also. And obviously, there's other folks who have worked on early development, like fetal and those sorts of things. And the Sanger, among others, have been working on that in mouse and human. And we're kind of leaving that to them to do because they're doing a fine job, and we're focusing on what happens after birth.
Okay, great. I think we'll take one last question from Carmen Gerlach. In your parabiosis experiment, did you establish the atlas by taking into account whether the RNA comes from cells of the older or younger partner, having either stayed in the original host or crossed over to the partner using congenic markers or alike?
Yeah, that's a very interesting question. So they share the blood, but not the solid tissues. And so, most of the analysis focuses on the solid tissues. So we're fairly confident that we know which mouse they come from. But obviously there's ambiguity there with any cells of the immune system in the blood, which come from either. And in this case, they weren't separately labeled.
Okay, thank you very much.